Good day, this is Professor Will, CBMD PhD. Today, Saturday, July 13, 2019. It is 2:03 seconds Eastern Standard Time. And this is another research reading on pathophysiology. Hope you enjoy listening. New technology improves atrial fibrillation detection after stroke. Date July 12, 2019. Source Michigan Medicine, University of Michigan. Summary. It is important to determine whether stroke patients also experience atrial fibrillation. Monitoring technology can make the process easier and more accurate. A new method of evaluating irregular heartbeats outperformed the approach that is currently used widely in stroke units to detect instances of atrial fibrillation. The technology, called electrocardiometrics, goes further than standard cardiac telemetry by examining large amounts of telemetry data in a way that is so detailed it is impractical for individual clinicians to attempt. Researchers validated the use of their technology in a clinical setting, finding the electrocardiometrics was an accurate method to determine whether a stroke survivor had an AFib. A crucial metric. After a stroke, neurologists are tasked with identifying which risk factors may have contributed in order to do everything possible to prevent another event. That makes detecting irregular heartbeat an urgent concern for these patients. Atrial fibrillation is a very important and modifiable risk factor for stroke. Importantly, the electrocardiometrics identification method was highly accurate for the 212 patients who did not have a history of AFib. Researchers say this group is most clinically relevant because of the importance of determining whether stroke patients have previously undetected AFib. When a patient has AFib, their irregular heartbeat can lead to blood collecting in their heart, which can form a stroke-causing clot. Many different blood thinners are on the market today, making it easier for clinicians to get their patients on an anticoagulant they'll take as directed. The most important part is determining AFib's presence in the first place. Much needed improvement. Researchers say challenges persist in detecting intermittent AFib during stroke hospitalization. More accurate identification of AFib should translate into more strokes prevented. Once hospitalized in the stroke unit, patients are typically placed on continuous heart rhythm monitoring. Stroke neurologists want to detect possible intermittent AFib that initial monitoring like an electrocardiogram or ECG would have missed. Because a physician can't reasonably review every single heartbeat, current monitoring technology flags heart rates that are too high. The neurologist then reviews these flagged events, which researchers say could lead to some missed AFib occurrences or false positives in patients with different heart rhythm issues. In contrast, researchers' electrocardiometrics converts two-dimensional signals from the ECG into a three-dimensional heat map that allows for a rapid inspection of all collected heartbeats. Researchers say this method permits fast, accurate, and intuitive detection of cardiac arrhythmias. It also minimizes false positive as well as false negative detection of arrhythmias. We originally noted 5 false positives and 5 false negatives in the study, but expert review actually found the electrocardiometrics was correct instead of the clinical documentation we were comparing it to. More applications. The research lab also recently demonstrated the usefulness of the electrocardiometrics to differentiate between AFib and atrial flutter. In addition, the lab has shown the ability of electrocardiometrics to capture reduced heart rate variability in critical care patients. Researchers say they envision electrocardiometrics technology will one day be used to assist the detection of all cardiac arrhythmias online or offline, and side by side with the use of ECG. I believe that sooner or later electrocardiometrics will be used in clinical practice to benefit patients. Good day, this is Professor Will, CBMD PhD. Today, Saturday, July 13, 2019. It is 2:37:53 seconds Eastern Standard Time, and this is a continuation of my pathophysiological research. So let's hop to it here. Novel nanoparticles deliver CRISPR gene editing tools into the cell with much higher efficiency. Researchers use lipid nanoparticles to deliver CRISPR-Cas9 gene editing tools for potential treatment of hyperlipidemia. Date July 12, 2019. Source Tufts University. Summary. Researchers have developed a significantly improved delivery mechanism for the CRISPR-Cas9 gene editing method in the liver. The delivery uses biodegradable synthetic lipid nanoparticles that carry the molecular editing tools into the cell to previously alter the cell's genetic code with as much as 90% efficiency. The nanoparticles could help overcome technical hurdles to enable gene editing in a broad range of clinical therapeutical applications. The CRISPR-Cas9 gene editing system has become a powerful research tool uncovering the function of hundreds of genes and is currently being explored as a therapeutical tool for the treatment of various diseases. However, some technical hurdles remain before it can be practical for clinical applications. CRISPR-Cas9 is a large molecular complex containing both a nuclease Cas9 that can cut through both strands of a targeted genomic sequence and an engineered single-guide RNA sgRNA that scans the genome to help the nuclease find that specific sequence to be edited. Since it is a large molecular complex, it is difficult to deliver CRISPR-Cas9 directly into the nucleus of the cell, where it can do its work. Others have packed the editing molecules into viruses, polymers, and different types of nanoparticles to get them into the nucleus, but the low efficiency of transfer has limited their use and potency for clinical applications. The lipid nanoparticles described in the study encapsulated messenger RNA, mRNA, encoding Cas9. Once the contents of the nanoparticles, including the sgRNA, are released into the cell, the cell's protein-making machinery takes over and creates Cas9 from the mRNA template, completing the gene editing kit. A unique feature of the nanoparticles is made of synthetic lipids comprising disulfide bonds in the fatty chain. When the particles enter the cell, the environment within the cell breaks open the disulfide bond to disassemble the nanoparticles, and the contents are quickly and efficiently released into the cell. We are just starting to see human clinical trials for CRISPR therapies. There are many diseases that have long been intractable for which CRISPR therapies could offer new hope. For example, sickle cell disease, Duchenne muscular dystrophy, Huntington's disease, and even many cancers. Researchers' hope is that this advance will take us another step toward making CRISPR an effective and practical approach to treatment. The researchers applied a new method to mice, seeking to reduce the presence of a gene coding for PCSK9, the loss of which is associated with lower LDL cholesterol and reduced risk of cardiovascular disease. The lipid nanoparticles are one of the most efficient CRISPR-Cas9 carriers we have seen, says researchers. We can actually knock down PCSK expression in mice with 80% efficiency in the liver, suggesting a real promise for therapeutical application. Today, this is Professor Will, CBMD, PhD. Today, Saturday, July 13, 2019. It's 3:10:39 seconds Eastern Standard Time. This is a continuation of my scientific research on pathophysiology. So let's hop to it here. C. difficile resists hospital disinfectant, persists on hospital gowns, stainless steel. Date July 12, 2019. Source American Society for Microbiology. Summary: surgical gowns of stainless steel remain contaminated with the pathogen Clostridium difficile even after being treated with the recommended disinfectant, according to new research. The spores of the bacteria were able to grow after discontamination. This shows that spores are becoming resistant, and we need to reconsider how we decontaminate and employ hygiene measures in hospitals. C. difficile infects roughly 500,000 Americans annually, killing 29,000 new strains that are responsible for hard-to-treat cases of severe illness. Symptoms can range from diarrhea to fever, rapid heartbeat, inflammation in the intestines, and kidney failure. This pathogen commonly affects older adults in hospitals and in long-term care facilities. The motivation for the research was a case in an American hospital in which gowns were suspected of contributing to transmission of C. difficile. The gowns were found to be contaminated with the deadly 027 strain of C. difficile. The researchers examined the ability of C. difficile to adhere to and subsequently transfer from hospital surgical gowns by applying spores in sterilized water at a concentration of 1 million per milliliter directly to the surgical gowns in liquid for 10 seconds, 30 seconds, 1 minute, 5 minutes, and 10 minutes before being removed and discarded. That methodology was designed to mimic transfer of infectious bodily fluids in a clinical setting in order to assess the potential for transmission to patients. The numbers of spores recovered from gowns did not increase with contact time, suggesting that the spore transfer between surfaces occurred within the first 10 seconds of contact. The single-use gowns are ineffective at trapping spores within their fibers and preventing downward transmission of spores as demonstrated by spore recovery from the gowns, according to the investigator. This highlights the importance of ensuring that single-use surgical isolation gowns are used appropriately in infection prevention and control. Important example: that personnel don the gowns upon entering and dispose of them when exiting a single room to prevent spore transmission and incidence of C. difficile infection. The gowns were ultimately treated with disinfectant containing 1000 ppm chlorine for 10 minutes. The disinfectant failed to clear the gowns of C. difficile. That showed that the gowns can pick up and retain the spore. The spores on the stainless steel and vinyl flooring also remain viable after treatment with the disinfectant. Due to this resistance, it may be prudent to reconsider how much biocide we use currently, and to ensure infection control is standardized. This work can be applied to hospitals anywhere in the world, and should help inform future guidelines on infection control and biocide. Good day, this is Professor Will, CBMD PhD. Today, Saturday, July 13, 2019. It's 3:37:31 seconds Eastern Standard Time. And this concludes today's research on pathophysiology. Hope you enjoyed listening. Have a great day.